The classical complement pathway is a part of the innate immune system. This pathway targets viruses and bacteria. It does so by providing a cascade of protein reactions that via the protein's direct binding to the antibody. The alternatives to the pathway are the lectin pathway and the alternative pathway. The result of this pathway is the formation of MAC as well as opsonization initiation. Here we have an antibody just roaming around waiting to spring into action, when suddenly a foreign pathogen enters, triggering the antibody to attack. The antibody lures the antigen to its Fc region where a conformation change occurs. This conformational change allows the C1 complex, consisting of components C1R, C1Q, and C1S to bind and be activated. After it is bound, calcium reacts with it to activate C1S and C1R. Next, C4 and C2 are recruited, and in the presence of C4, C2 splits into C2b and C2a, which diffuses away. C4 is also broken into subunits called C4a and C4b. Next, C3 convertase is formed from the binding of C2b and C4b. The C3 convertase then interacts with the C3. C3 can now be broken into C3a and C3b. C3b then binds to C2b and C4b to form a new complex, the C5 convertase. This convertase cleaves the C5 into C5a and C5b. C3b then serves as a signal for macrophage uptake or opsonization. C5b then activates C6, C7, and C8. And those form a new complex that recruits C9. The new complex forms the membrane attack complex or MAC. MAC creates pores within the cellular membrane as a means of lysing a cell and killing the pathogen.